Noon and good evening. My name is John DeBella, Vice President of Marketing and Sales, and on behalf of Simulations Plus, I welcome you to today's webinar, Lead Hopping and de novo Design with MedChem Studio. Michael Lawless, Team Leader, Chem Informatics Studies, will be presenting today. Joining Michael and myself are Anu Sharma, Senior Marketing and Sales Manager, David Miller, Team Leader, Discovery Chem Informatics, and Bob Clark, Director of Life Sciences. An opportunity to ask questions will follow Michael's presentation today. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you're using a telephone to listen to the call, please be sure to enter the unique audio PIN displayed when you join the call. When you enter your audio PIN, this enables us to unmute your line so that you can be heard. This webinar is being recorded for playback at our website, www.simulations-plus.com. It is now my pleasure to turn the presentation over to Anu, who will provide a brief introduction to our company. Anu?
Thank you, John. Hello, everyone. So I'll start with a brief introduction of the company. We were founded in 1996, and we are headquartered in Lancaster, California, which is an hour-and-a-half drive from Los Angeles. Ours is a publicly traded company with 25 employees. We are devoted to developing modeling and simulation software. On the right-hand side, you see a product portfolio. GastroPlus is our flagship product, which is used for simulation of in vivo absorption, pharmacokinetics, pharmacodynamics, and DDIs. ADMET Predictor is another very popular product which is used for the simulation of admetox properties and for building and training models. MedChem Studio, which you will be learning more about today, is a product that is used by the medicinal chemists for data mining and in silico drug designing. DDD Plus is a tool for formulation chemists for the dissolution experiments. MedChem Designer is for the drawing and the sketching of chemical structures. So as you see that Simulations Plus offers tools that are used at the various stages of the drug discovery programs. ADMET Predictor is used by the biologists and the chemists in the earlier stages of the drug discovery to the development stage. MedChem Studio and the MedChem Designer are used for lead hopping, lead identification, and lead optimization. And GastroPlus and the DDD Plus are also in the drug development stages. Next, I'm going to be introducing you some of the new and the advanced features in ADMET Predictor and MedChem Studio. So the latest upgrades that we have introduced in ADMET Predictor 6.5 are we have a better graphic user interface, and also there have been enhancement in the general program applications, and we have also introduced several new models. So one of the biggest change has been that we have outgone the exchanging and unlocking of the codes over the phone. We now use the FlexNet Manager, which is a server-based license management system in the industry. And this has been used in the industry for the last 20 years. So most of our customers are used to running it on their local networks. You can now predict with confidence. We have introduced the prediction confidence scores, which is the probability of correctness of prediction in selected classification models. New predictive models that have been introduced is the air-water partition coefficient model, which is the Henry's law constant. Then there is the skin permeability model also. And we have rebuilt 10 models of Ames mutagenicity. We have also expanded and improved to the graphing features, the minor 3D graphing. And we have also 18 new site-specific SIP-mediated models. In the new features in the MedChem Studio 3.5, we have made several improvements, did several bug fixes, and we have many new features. The ones that I will be introducing here are just to highlight some of the important ones. So one a very important feature is the R group analyzer. So now you have position tabs for each of the R groups. And there are columns for the number of subsequent occurrences, and also there's a column for property average and distribution. On the left-hand side, you see is a window that is used for resizing the buttons. And on the substituent column, if you click, it's going to open the structure viewer window, which will also have the molecular attributes. We have also upgraded the Miner 3D interface, so now you can see the data in a variety of different graphs, charts, bar graphs, and so forth. We have also maximized the extraction information in MedChem Studio, so the information that is otherwise difficult to detect by the human eye is now possible in the MedChem Studio. We have introduced the URL links in the spreadsheet, so you could open up any web page associated with the molecule, as well as you can open up MedChem any document, the MS document. So in this screen, as you see, we have opened the PowerPoint presentation by just clicking on the link. We also have the template database, so the users can customize the way the molecules are drawn, and they can store it as they like, and if they do not like the feature, they can turn this off as well. For the matched molecular pair screening options, we have several upgrades in this feature as well. And again, for the matched molecular pairs, you can order pairs in the molecular pairs tab by similarity to a user-defined scaffold. Again, grouping in the matched molecular pairs by the common transformation is possible, so you could identify transformation associated with consistent property changes. Last but not the least, MedChem Studio has a variety of features. So there have been a number of articles lately in which the various organizations have used the matched molecular pair approach for the areas of investigation, such as ligand potency, aqueous solubility, and as you can see on the right-hand side of the screen. So MedChem Studio offers this features as well. So I'm going to hand over the presentation to John to go over in more details on the MedChem Studio now. Over to you, John.
Okay, thank you, Anu. It is now my pleasure to turn over controls here to Michael Lawless, who will be leading the presentation on lead hopping and de novo design with MedChem Studio.
Thank you, John. Good morning and welcome to our webinar. Thank you for attending. This next slide simply shows an overview of the analog generation and lead hopping process. So a lead molecule can come from many sources, including competitors' patent or internal screening. So some of the reasons for hopping away or developing analogs of your lead molecule is to circumvent a patent, for example, explore SAR and hopefully improve potency. You can also generate new molecules that hopefully will have improved bioavailability and reduced toxicity. So this webinar is focused on lead hopping and analog generation. So once you've generated these molecules, we recommend reevaluating target potency either via a QSAR model or docking or both. We also recommend computing ADMET properties, and of course we have a nice program, ADMET Predictor, for computing various ADMET properties, and then filtering your list of potential candidates in order to get down to the synthetic candidates that you'd kind of hand over to the synthetic chemist. So the talk is basically goes in 4 sections. So in the first section I'll discuss generating novel and analog molecules using transformation-based reactions. In the second section, I'll demonstrate molecular modifications using fragment-based replacements. So with this, the first step is to generate fragments from a database of molecules and then use those fragments in assembling them together to create a new compound. In the third section, I'll discuss R-table explosion. So basically, you would have a set of active compounds around a common core, and you wish to enumerate all the unfilled space in that chemistry space. Excuse me. And then in the final section, I'll talk about creating and searching virtual libraries. So the first thing to do is to create libraries, and one way to create the libraries is to enumerate combinatorial chemistry libraries. Then you add these to our MedChem Studio proprietary formatted database, and then use matched molecular pair analysis to compare your lead molecule to all the molecules in the database, and use a similarity metric to find the most similar compounds to your query molecule. This next section discusses these transformation-based reactions. So if a medicinal chemist gets a compound such as this, there are some rule of thumb modifications he would typically make to the compound. So this compound contains a carboxylic acid functionality, and we predict the pKa of that to be about 3.83. So you might want to substitute that with a tetrazole, which has a pKa of 4.88. So it's a little bit higher pKa, but maybe this will make the compound more permeable to cells and other areas where it needs to permeate through a membrane. Another transformation might be to convert the carboxylic acid into this hydroxyisoxazole. Here we predict the pKa to be 7.3. Remember, though, that a pKa of 7.3 means that more than 50% of the molecule is deprotonated at a pH of 7.4. You also might take this amide bond and simply flip it, or to create the reverse amide, and then finally replace the amide with a hydroxy group, which still would contain the hydrogen bond donor and acceptor groups. So there was a paper published in 2006 by a group of Abbott, and they created a program called Drug Guru, and we've kind of implemented a similar system within MedChem Studio. So our transformations are encoded as smirk strings. So for example, this transformation searches for carboxylic acids in the molecule and converts them to tetrazoles. We've created a default file of 120 transformations that was developed in collaboration with medicinal chemists. Each transformation in the file has a category, and we can enable or disable specific categories. We can customize the transformation file in order to exclude some transformations and include others. So you'd simply probably make a copy of that file and then just remove ones that you don't want to and include others at the bottom. And of course, you can create just your own file with specific transformations for your particular task. A major feature also is the ability to remove undesirable products, and we use what we call a filter file that's a very versatile file for removing things based on property predictions or based on substructural moieties. This next slide shows just the first few lines of our default transformation file. Remember, there's 120 of these transformations. Each transformation has an attribute describing the particular transformation, and this is output to the file of transformed molecules, so that you can easily see what transformation was responsible for creating that particular molecule. The next header is a reaction class. This enables us to segment a portion of the reactions to a particular class. So all these that I've listed here are in a change functional group, and you can enable or disable that particular transformation. We have a number of transformation options within the program, so we can specify the maximum number of total transformations for any one molecule. We can also specify the maximum number of times that a single reaction transformation is applied, and then finally specify whether the reaction transforms are applied in all possible orders in the file. So I'm going to show 3 examples here, cartoons, where we have 4 transformations, A, B, and C, of a primary molecule shown at the top. And all these transformations are allowed within the molecule. In other words, the molecule contains these moieties to be transformed. So if you specify a maximum of total of 2 reactions, 1 application per reaction, and not all orderings, you would produce 10 compounds. So in the first step, it would take this compound and produce these 4 compounds that would be transformed via A, B, C, and D. Then it would take each one of these molecules and potentially apply a secondary transformation. So you would get the molecule shown here that occurs from transforming it first via A and then transforming it via B. Now, you don't get A transforms twice because we specified a maximum of 1 application per reaction. So you also don't get BB or CC and DD, so we end up with 10 products. This next example shows a maximum of 2 total reactions and 2 applications per reaction, and then not all ordering. So here we are creating AA, BB, CC, and DD for an additional 4 products by allowing an application to occur twice. And then finally, if we go for 2 reactants, 1 application, and on all orderings, we produce these 16 molecules here. So we produce A, etc. Again, AB and BA would be formed, but only if reaction A is affected by reaction B. So what I mean here is if, for example, you had a carbonyl and the first transformation maybe transformed it into a thiocarbonyl, and then B transformed it back to the original molecule. That would only occur if the 2 reactions are different. Okay, so let's hop into the program here and bring up MedChem Studio with some starting structures. So in the first case, I'm just going to show converting this molecule using a single reaction called a reverse amide. So it works on the highlighted molecule, and I would just go down to the reactions pull-down menu and select combinatorial transformation. Okay, so starting at the top of the dialog box, we're going to write the input molecule to the output file so that we can kind of identify it and compare it. Then I'm going to go and browse or specify just 1 single reaction here that I've saved, and this is this reverse amide transformation. So it's a single transformation in the file, and what it does is perform the reverse amide. I'm going to apply it a single time for these defaults, and then there's an add filters. Now this is where you'd specify your product filtering, but there's also something that says reject products already contained in the spreadsheet. I'm going to turn that off for these examples, and then I need to output the molecules or specify an output file, and I'll just put that on my desktop for right now. Then I click OK. It goes out and performs these transformations and shows them in another session of MedChem Studio. So here's the original file, and then the 2 products produced are transforming this amide first, and then in the second molecule transforming this amide. So that's just a simple application of 1 transformation and shows you how MedChem Studio would work. Okay, so now let's show a second example of naphthalene. And in these next few examples, I just want to show, imagine if you had a core molecule and you wanted to apply transformations to that core. And just want to illustrate some of the transformations in the file and what products are produced. So again, I'll go down to the transformations. Here I'm going to specify our default file within the MedChem Studio tree. And that's stored in the data, and it's called molecular transformation. So I just double click on that. Again, we have the output file, and I'll just click OK. up will come the transformations for naphthalene. So we have naphthalene first, and then this first pair of molecules here is from applying the transformation aromatic carbon to nitrogen. So within naphthalene, you really only have two unique positions to put the nitrogen, so you would make a quinoline and an isoquinoline. Then the next four structures are from adding an amide to the one of the rings. We also have a couple that would form an indole and an isoindole. Okay, so for this quinol, I want to illustrate again some reactions. I'll just use the defaults again. Click OK. And then I wanted to show a specific transformation. So I'm going to search the product confirmation for the word pyridone. And you see that gives 2 hits, and I'll just take that to the top of MedChem Studio. So here we show the initial molecule, and then these transformations take this nitrogen and move it to the bridgehead position to produce this one, and then move it to the exterior position here to form this molecule. So those are just a couple more examples. Then with benzimidazole, I'll perform additional transformation. Here I'm going to add a filter file that I've created, and I'll briefly describe the filter file. So let me first go to the directory and specify the file, and then I'll show you what's in that file. So I just added this to a Word format, or Word formatted file. So we call this I call this drug-like substructure.cqf. So it's a filter to remove files containing non-drug-like fragments. And I'm always kind of updating this file as I do searches and look at molecules and say, well, that's not very drug-like, so I'll encode a smart string to maybe remove that. So this first query removes non-standard atoms. So if you have anything other than this set of atoms in the molecule, it will eliminate that. And of course, users can modify this. You may want to include iodine. I personally exclude iodine in my products. You might also want to include boron, etc. And then these next few queries are to limit some of the aspects of the molecule. For example, if the ring size gets above 7, I want to eliminate that. If there's more than 10 rotatable bonds, I would eliminate that. More than 5 hydrogen bond donors or 10 acceptors, etc. Then as we move down, there are some specific filters such as removing aldehydes, sulfonates, hydrazines, etc. So you can add, create your own file and add any filters that you want. And of course, I took some filters from papers. So there's a paper from Jonathan Bell called Payne's filters, and these are some specific filters for that. So just go ahead and click OK for this benzimidazole, and just want to show again a few of the products that are produced. So you get the azo benzimidazoles, nitrogen in the phenyl ring, some benzoisoxazoles, benzothioxazoles in this indazole compound. Okay, so let's say there's a transformation that's not in the file and you want to customize the file to include that transformation. So you would simply create a transformation a file, and I've already created this transform1.cft. So this transforms a pyrazole to an imidazole. So here's the query on the left. So it's nitrogen, nitrogen, carbon, carbon, and then these :1, :2, etc. identify the atoms in this molecule and then map them to the product. So basically, I'm just converting this nitrogen to a carbon and this carbon to a nitrogen to form the imidazole. So I would need to go to the combinatorial transforms, specify the transformations, go to my shortcuts here, and find this transformation here, transform 1, sorry. Then go ahead and click OK. and up comes 2 molecules. So in the first case, it's taken this nitrogen over here and moved it over to the bridgehead. In this bottom case, it's moved this nitrogen over to here. Now, this is substituted with a hydrogen, so you can just remove that hydrogen and we keep the valences correct. Now, if that was substituted with a methyl group, then you wouldn't be able to move this nitrogen over here and still retain the aromaticity. So I added that. You don't see it in the filter, but that was added as a filter to eliminate that particular product. Now let's briefly go back into the slides. And so now I want to demonstrate applying transformations to Viagra. So I created a custom file that would take pyrazole to imidazole transformation, and I just added it to the bottom of all the standard transformations, and then save that as a new file. So I'm going to specify a maximum of 2 reactions per output molecule and a maximum of 2 for any single reaction, apply all the reactions in all possible orders, and again have the substructure filter on. So I highlight sildenafil, go up to the combinatorial transforms, browse to the directory. So this is the standard molecular transformations plus 1 additional one. Simply open that up, specify 2 for the maximum here. The filter file's already been applied, so I'll just click OK, and then it goes through and it performs these transformations very quickly, and it's also filtering out the products from the drug-like filters. So this is only applying 2 transformations. If you apply 3, of course, you'll produce more products in your output file. As always, it's important to then filter in the end for ADMET properties. Okay, so we've produced a couple thousand molecules here, and what I want to show is just search through here and show that from this transformation, we have come up with Levitra. So I want to show that we've transformed Viagra into Levitra. So I'll simply go into the MedChem Studio output file, and then I'm going to query for Levitra using MedChem Designer. So open up a substructure of Levitra, paste it back into the query here, and then you'll see that Levitra is found in there, and I'll just pull that up to the top to compare it. So in this molecule Levitra, we've moved this nitrogen over to the bridgehead, and then we've added a methylene group between the methyl and the nitrogen group, and again we see the 2 transformations here. On this next slide, I show the property distribution for the 2,400 or about 2,400 compounds that were produced, and in each one of these plots, there's a bin with a red line, and that indicates the bin that holds our query molecule. And as you can see in all these distributions, we get a fairly good Gaussian distribution around the query molecule. So log P, we end up kind of on both sides, which is the kind of thing you want in your output. So the TPSA also transforms very nicely and looks like a Gaussian distribution. For the solubility, we've actually got a little bit more compounds with higher solubilities, but we've also increased the molecular weight a little bit here. Okay, so another feature that kind of combines 2 of our tasks is using transformations to maybe increase an ADMET property. So I'm going to demonstrate using matched molecular pair analysis to generate transformations and then apply those transformations to a molecule with low solubility in order to hopefully create compounds with higher solubility. So we had a webinar a few months ago on matched molecular pairs. Within that, I showed a database of molecules. This one contains about 3,000 compounds with experimentally measured solubilities. So this is this log S molar column. Then we simply go to the pairs tab. We want to generate pairs. So here what we're doing is we're going to compare each molecule to each other and see how large the change is, and these are these values up here. And we're going to specify that I only want to see changes where the solubility is increased by an order of magnitude based on these transformations. So I would click OK, and then up in the molecule pairs would be the pairs. So I've already done this on this particular set. So here it's generated about 4,400 rows of transformations, and in each one of these transformations the change in solubility has gone up by at least an order of magnitude. So I can sort on these columns. These are the ones, transformations that are only order of magnitude. The ones at the top, this transformation increases the solubility of this molecule by 6 orders of magnitude. So I could highlight all these reactions and go down to generate reactions. Now what this is going to do is analyze each one of these reactions and create a smirch string for that particular transformation. These two options here control how far away from the modification should we include, or how many atoms should we include away from the transformation. And I just specified 5 and then specified an output, and it wrote all the transformations to the output file. So now I have transformations that will increase the solubility of a compound, and so I'll go back down to combinatorial transforms and specify that file. So this is this increased solubility transforms that was created for this. I'm only going to use one transformation on each of these molecules, again applying filters. And I would click OK to create those transformations. So I'll just go ahead and click OK. And this is only a limited number of molecules since we're only doing one transformation apiece. Once this comes up, I'll open up the other file that I had created, and where I actually added predicted solubilities to each one of these compounds. So I don't think I've got it opened here. Oh, I do have it opened here, sorry. Okay. So this is the same 61 transformations, but I've added predictions of the water solubility. So here's the initial product, and if I sort on solubility, you'll see that not all of these transformations actually increase the solubility. Here's the original molecule, and then there's about 6 or 7 compounds where the solubility was not increased. We can make a distribution plot of these solubilities over here, and you'll see that our initial molecule, which is a mGlu R2 positive allosteric modulator with a solubility of 10 nanomolar, would be down here in the distribution, and then all these other compounds down in here would have increased solubility over this particular compound. So again, I'll sort in descending order, so you see things like adding a carboxylic acid to various positions on the scaffold, also converting the these aromatic rings to heteroaromatics to also increase the solubility for that. So that was an application of applying matched molecular pair analysis to a particular compound in order to increase the solubility. And you could do this with other transformations, maybe to increase log P, etc. Now, moving to the second section, I'll be discussing generating and assembling fragments. So there was a paper published in 1998 from a group at GlaxoSmithKline, which they implemented some recap rules, which are retrosynthesis combinatorial analysis. So this is an analysis for fragmenting the molecules and then assembling those fragments back to create new molecules. So in the first step, we have to generate the fragments and break them into smaller pieces. So we have 2 files that specify the fragmentation rules. One are these recap rules. Another one is called MedChem specific, which will break up the molecule into a lot smaller pieces than the recap. These recaps are based on synthetic rules. Then when you generate the fragments, you can output the file to a CTK file and then use this CTK file to recreate or to create new molecules. The program also has the option to output SD or SMILES files where the attachment points are tagged with at the cleaved position, and this can be used maybe in other programs. Okay, so one thing we did was to take a database of known drug molecules and apply these chem-specific fragments to generate drug fragments, and then you can take your query molecule and build new molecules using this file of drug molecules, and you ensure that every fragment in that drug file is from an actual drug. So when you assemble these together, you can create new compounds, either replacing the scaffold in your query molecule or the side chains. So here's just some fragmentation results based on the retrosynthesis analysis. Again, we have our diamide, and so the recap rules would just break this into 3 pieces, kind of 2 side chains and a scaffold. Our chem-specific file would break this up into much smaller pieces, so you'd have the thiophene, the carbonyl, the amine, the phenyl group, and then the quinoline as the fragments here. So let me again pop into MedChem Studio. This is the file I need to close. Okay, and I just want to show these are the recap rules. So we applied the recap rules, and you'll note in this is the fragment that would be pieced together. You see that the attachment points are marked within this file. Okay, so if we take our diamine and define this as the scaffold, and then go up to the assemble fragments dialog box, and here I'm going to specify the input file is the recap file that I just picked. So I'm sorry, I pushed the recent documents instead of my desktop file. So let me just take a second here. Okay. Okay, so here's our recap fragments, specify an output file, and I'm just demonstrating here that from either of these positions you can recreate the original molecule. Again, I have to make sure that it doesn't, it allows this initial molecule here in the output file. So this is the generated file using the recap rules. So just to illustrate that from the recap we reproduce this, and then also I'm going to specify the other file that was created from the chem-specific transformation or fragmentation. So again, this would apply these rules, but it builds them up and reproduces the original molecule. And then lastly, I'm going to illustrate taking that diamide again and using the default rules within this to replace this amino acid kind of scaffold here. So I go down again to reactions, assemble fragments. This time I'm going to go to the default file within MedChem Studio. And I want to use drug fragments chem-specific. And specify filters. Now, what I didn't mention on this is for the output molecules, we have 2 options to control the minimum and maximum product count as a percentage of the original molecule. So this keeps the molecules fairly similar to the input molecule. If you want to get very much larger or a little bit smaller molecules, you could change these to defaults. Again, I'll go over to the apply a drug substructure rules to these, and then go ahead and click OK, and then OK again to produce some of the transformation. So again, it goes through and produces, builds up the molecule, and then filters them based on the filtering rules. So here we get 31 new molecules. So you have this amino acid type scaffold in each one of these molecules. Here we have something like pyrazine. So these first ones are saturated rings, which would change the geometry from the query molecule. And then we have more molecules that are like the pyridone as the core, pyrimidine as the scaffold, fluorophenyl, and then heteroaromatics such as the isoxazole and the thioisoxazole, etc. So assembling fragments is a nice way to replace the core with drug-like fragments. Now returning to the slides, kind of talked about that already. So in the next section I'll briefly discuss R-table explosion. So this is a way to generate analogs of a set of molecules. So in this case we've taken an isoxazole core. Again, this is from M-glue R2 positive allosteric modulators, and we analyzed the data, came up with this scaffold, and then there are the R1s are listed as the rows, and the R2s are listed in the columns, and then each place you see a circle, that indicates that this molecule is formed, and the color is relative to this scale down here. So these are PEC50 values, so 4.52 is between 10 and 100 micromolar, 7.09 would be sub 100 nanomolar. And there's a lot of cells in here that are unoccupied, so this is unexplored chemistry space. And we can just use a feature within MedChem Studio to explode this, to enumerate it, and create additional virtual molecules to kind of fill out the chemistry space here. So let me go to the oxazoladenone scaffold. So we're in the classes tab. We've already defined the R groups, and then our table, and then we'll go to the R groups. This is identical to the slide I showed. So we'll go down to the R groups, pull down menu, and select explode products. So we need to specify an output file. Again, I'll just put this on my desktop and save that. We have a maximum number of products that are produced, and I'm going to remove any products that are already contained in this spreadsheet. We also have options of selecting R groups and disabling them from the explosion. So I could enable or disable specific ones. I can query for specific R groups, maybe to remove and unenable. So again, I just go ahead and push OK. This performs the combinatorial explosion and creates 380 compounds in this case. And I'm just going to quickly put them in, combine them all into one class, define the scaffold, again by circling it over here, and then go into the R group pairs menu, and now you see that all of the chemistry space is filled, except for these column and row here, and these are the original molecules that were in the previous spreadsheet, so there's no need to recreate those here. Now in this last section, I'll discuss creating and searching virtual libraries. So this is kind of an outline of the overall procedure. So the first thing is to identify sources to add to the database. So one source is to create your own combinatorial chemistry reactions, and MedChem Studio has a nice way for enumerating combinatorial chemistry libraries, and I'll demonstrate that, and then also show a few reactions that I created and put into a database. Of course, you can look at various suppliers databases that are in the public domain, or you could use your corporate database, of course, for the virtual libraries. So Zinc is a free database for commercially available compounds specifically created for virtual screening. And here's the reference for Zinc. So once you've identified various sources and downloaded the SD or SMILES file, you can process these into a MedChem Studio formatted database, which allows you to select the attributes that are stored in the database, and then it lists each individual file separately, and you can search in either individual databases or a list of databases together. And then finally, we need a similarity metric to identify molecules that are similar to the query method, and I'll talk about that on a subsequent slide. So there was a nice paper published in 2011 in which they, in their supplemental information, they show about 50 combinatorial chemistry reactions that are very robust for combinatorial chemistry synthesis. They also included the SMART string to search for the reagent classes, and then the SMERC string that will transform the reactants into the product. So this is a nice paper to get and the supplemental data and then encode these libraries within MedChem Studio. So the first item in the process is to search for vendor databases for amenable reagents. So for example, you want to find nitriles and these acyl hydrazines compounds. Then once you perform the searches for these reagents, you want to kind of look over that list and filter for unstable or undesirable reagents. So over on the right, I show a nitrile that contains 2 nitrile groups, and typically you wouldn't want to put this in a reaction, so we would eliminate that. Similarly, if a compound contains 2 reactive functionalities for the reaction, you would want to eliminate that. So I eliminated both those type compounds. Then I combined both of these reagents into MedChem Studio, created classes for each reagent types, and then I need to specify this reaction. So we have 3 different options for specifying the reaction. I'll demonstrate just sketching the molecule, the reaction in ISIS straw, and then mapping the reagents to the products, and then also you could use a SMERC string. And then finally just pushing the enumerate button to enumerate the library. So I'll go ahead and demonstrate that right now. So of course, I pre-processed a lot of these for the demonstration. So here we have this first reaction. So we have 2 reaction classes. The first one are these nitriles. There's 730 compounds in there. And then we have these acyl hydrazines, and there's 253 in this particular class. So we'd simply go down to the reactions, perform reaction, and then here's where we would specify our SMERC string. So we have a nice shortcut to produce that SMERC string. So you can just sketch the reaction in ISIS straw, as I've done here, and then use this tool here to map the atoms in the reagents to their position in the products. And sometimes it takes a little bit of modifying this to get exactly the products you want. Typically when you perform the reaction within MedChem Studio, you want to search for this core and make sure that all the products contain the core molecule. So here I would just highlight this, go up and copy that into the buffer, and then go back into MedChem Studio and paste that. And MedChem Studio converts that reaction into a SMERC string shown here. Then I'm going to specify the output file. Again, I'll just put this on the desktop as a CTK file and save that. Now, for this demonstration, I'm only going to explode 100 products, but you can see by combining 730 with 253, you'd actually produce closer to 200,000 structures. So just click OK. It enumerates this very quickly since there's only 100, and then you see this triazole in every product. So this is kind of interesting. This other triazole was actually in the reagent itself. Not sure if you want to keep all the ditriazoles, but you could specify a filter file to remove those. So now I'll just, the next slides just show a few examples of combinatorial chemistry reaction. Again, some of these are from this paper. So I already talked about the triazole formation. I also encoded this particular library, which creates benzimidazoles by combining a dianiline with either aldehydes, carboxylic acids, or esters. We can create quinazolones from combining this kind of amino acid that's orthosubstituted to amide to create the quinazolone. This particular reaction, the Buchwald-Hartwig, will produce anilines by combining aromatic halides with either anilines or primary and secondary amines. And then finally, I encoded library to create amino alcohols from epoxides and amines. So the next step, once I enumerated these libraries, then I applied my drug-like filter, and that created another SMILES file, and then I created a library from each one of the filtered files. So here I have about several examples. I have the triazole library that's got 173,000 compounds in it, the amino alcohols, the two sets of benzimidazole, about 2 million structures from this Buchwald-Hartwig reaction, about a little over 40,000 quinazolines, and then finally 6 million from a filtered zinc set. Okay, so once you've created these databases, then the next thing is to search them, and we have an algorithm called match molecular pair similarity that uses maximum common substructure. So essentially it finds a maximum common structure between two molecules and then lists the matches and the mismatches. So the metric for similarity is very chemically intuitive. It's just based on the number of matched and mismatched atoms and bonds, and we calculate the similarity based on the number of matches divided by the total of mismatches plus matches. So in this pair, there's 18 mismatches and 34 matches, so that would be 34 divided by 18 plus 34 to give a similarity of 0.65 for this example molecule. Now let me flip back into MedChem Studio. So I have a number of compounds here that I use as query molecules. All these molecules, except for this one here, are known drug molecules. I got them either from the World Drug Index or from drugs that were approved last year in 2012. So you'd simply highlight the molecule that you want to search and then go down into the query and screen databases using MMP. So in all these cases, I used all these libraries to search. I specified a minimum of 20 atoms and a Tanimoto of 0.7 for these, and then I imported the results back into MedChem Studio. And each one of these searches on my fairly old laptop took about 20 minutes using both processors. So you can very quickly search millions of structures in a fairly short time. So ENLITA was approved last year. Searching these various databases, I only came up with one hit, but it's an exact match. So ENLITA is actually in the zinc database. This next compound, Albagio, I got 98 hits, both from zinc and also from this Buchwald-Hartwig reaction. So we can sort on similarity. So this is the highest match. It comes from zinc, and so basically it's got most of the backbone of this molecule, but it's missing this hydroxy side chain, and that would probably be pretty important for activity, but it'd probably be worth maybe buying this compound and testing it in your screen. For the Buchwald-Hardwig reaction, remember we don't create amides. These are only aniline, so you'll never get the aniline produced a priori of this, but maybe this carbonyl is not important, so maybe it's be nice to test some of those molecules also. For Belvig, we got 83 hits. Most of them, I think all of them, were from the zinc database, and they're shown over here. We can sort on the similarity metric and see the best match pair. So shape-wise, this is a pretty good match. So we've got these exact 7 and 6-membered rings fused together. Substituent here, we replace the chlorine with the hydroxyl in this group. The methyl is replaced by another hydroxyl. One thing we are missing is a protonatable amine in this reaction, so maybe that's not your highest priority compound. Maybe down here, this one, which would contain a protonatable amine, might be a closer match and would be a better hit for this target. But you'd recommend testing all these. Okay. This next one is prazoxazole, and we got a number of hits, as you can imagine, from our triazole library. So the one with the highest similarity score is shown here. So we have this exact same side chain over here on the left side of the molecule. We've replaced the oxidizole with a triazole, but what you'll notice also is this side chain has a carbonyl, or an amide bond. So in the query molecule, this is protonatable. Here it's not, so you're changing the pH, pKa of the compound. This next one is a match I like quite a bit more. It's almost an identical match for most of the molecule, and then you still have a protonatable amine. Here you're substituting it with dipropyl groups. Mibrequin found several matches from the amino alcohol library. Of course, this does contain an amino alcohol itself, so this molecule is a very nice match for this, although we're missing this thiazole side chain. Now, some of the other hits specifically from the zinc database contain a heteroaromatic group up there, but some different substitutions on the other side of the molecule. So just going through and showing some examples. Let me skip this one and go to the N-glu R2 positive allosteric modulator again, and show a few hits from there. And these hits are coming up from the benzimidazole virtual library that we created. And these end up by default being sorted in descending similarity. So here we see a pretty good match for definitely the benzimidazole and the benzyl off of that. We've added a chlorine here, but you'll notice that we've taken this pyrazine ring and substituted it with a cyclohexyl. How important that is to the activity. You would have to test this. Potentially you could get some activity out of this and then make modifications in order to lead hop away from the query molecule. And then finally, I'm going to show some hits for aflexaquolone, and we got several hits within the zinc database, fairly good similarity, but we also got some hits from this quinazolone library that I encoded. So here we're creating the quinazolone and then a substituent off of there. Now, we are missing the NH2 in this particular reaction. But we have some very nice hits from this virtual library that we created. And I also want to mention that you can create virtual libraries around your lead molecules. Let's say your lead molecule came from screening and you want to create a very large library that contain that same core. So again, you could use MedChem Studio to enumerate that library and then perhaps do some match molecular pair searching to find the best matches to go ahead and synthesize. This last slide is simply a summary slide of the talk. So we'll discuss lead hopping and analog generation. So it can be used to get out of the scope of a patent, probe SAR, increase potency, bioavailability, or reduce toxicity. So I talked about the 4 different methods within MedChem Studio to find analogs or create analogs and lead hops. One is the transformation-based reactions, similar to Drug Guru. Fragment-based replacements are similar to the recap strategy. Our table explosion fills unexplored chemistry space in a series of molecules around a central core. The matched molecular pair searching can be used to search very large libraries. And then finally, you would want to apply ADMET property predictions to filter the molecules from any one of the techniques. And then finally, I want to acknowledge a lot of people that worked on this, Bob Clark and Dave Miller, of course, panelist here, and Dave is the team leader. Alexandra Mikos works for Dave in programming MedChem Studio, and then Walt Wotas is our CEO of Simulations Plus. And I'd like to thank you for your attention, and I'll turn it back over to John.
Thank you, Michael, for the excellent presentation today. We're now going to enter the questions and answers portion of today's webinar. Once again, you may either send your question using the questions pane on your control panel, or if you'd like to ask a question using your telephone, please use the hand-raising icon on your control panel and be sure to enter your unique audio PIN. There are several questions that we have in the questions pane that have been sent in, and so I'll start with the first one. And David and Bob, hopefully you're both unmuted for this portion as well. Can calls to external programs to return, for example, a docking score on enumerated compounds be incorporated into MedChem Studio?
The main way you would have to go about that right now is by way of hooking into something like Pipeline Pilot. But I don't, David, are the MMPA, is that set up for Pipeline Pilot batch use right now?
We do have a number of MedChem Studio features that are tied into Pipeline Pilot, but the match molecular pair, actually I think now we're talking more about the analog generation feature, the Drug Guru feature, and that is not yet been wrapped up into a Pipeline Pilot or NIME component, but that's something that we've sort of had on the plate to do. So right now, any of these sort of secondary calculations would need to be done as a post-processing step. You can see that the generation of analogs is, generally speaking, very fast. Doing it as post-processing is probably not too big an issue, unless you would like to sort of first kind of a screen, like a docking screen, and then based on that docking screen, decide whether or not to generate further analogs for a particular structure in sort of an iterative process. And so that's something that we've explored ourselves with the ADMET predictor predictions. Take us dual analogs of it, then predict solubility or other properties, and then based on those predictions, decide whether to generate more analogs of particular structures in order to drive.
Okay. Thank you, David. Thank you, Bob. Another question, any way of filtering products on the fly by calculated properties or MPO score?
I don't know if you can hear me. I think that's kind of the same issue as the previous question. It has to be a post-processing step, although, again, we're looking at a NIME or a Pipeline Pilot component more directly.
Okay, thank you, David.
Yeah, of course, the filter file that I showed has different properties in there that allow you to filter on the fly. So, for example, if you want to eliminate compounds that are created that contain 8-membered rings, you could remove that. Also, things like molecular weight, hydrogen bond acceptor. So there's a number of properties within MedChem Studio that it calculates internally. Other ones would be calculated from ADMET Predictor.
Okay, thank you, Michael.
Mm-hmm.
Another question, can R group axes be sorted, for example, by molecular weight or similarity score?
Referring to the R group pairs tab in particular. I believe that question came in when you were showing the R group pairs feature. I don't think you demonstrated the sorting of the two axes.
That's right. So let me just pull up that. Let's see. That's in R group explosion. Yeah, when you go into the R group pairs, then you can sort these in order of decreasing molecular weight. So both ways you can sort these. Now we cannot sort by similarity to a particular scaffold. David, that would have to be implemented. Is that correct?
That's true. There is an activity sort though. So if you right-click on a cell, on an empty cell in the interior, there's a sort there that allows you to try to arrange the entire table so that activity, any activity trend that is present would be brought out. It's probably not as easy to demonstrate on your very sparse.
Okay. So this is sorted in descending activity, I guess. So yeah, that's a feature we may want to add, similarity on the sidechain. Okay.
Thank you, Michael. Thank you, David. Another question, are ChemDraw reaction files supported for your combinatorial enumerator?
Yes, I believe so. I demonstrated ISIS draw, but ChemDraw also is supported. And David can maybe add a little bit to that. I don't have ChemDraw installed on mine, so I didn't demonstrate that.
Okay, Dave, anything to add to that?
Can you hear me?
Now I can.
I think there has to be dead silence in order for my phone to come through. It's using voice over internet. But with ChemDraw, you can export the ISIS-style RxN files. And so I'm not aware of a native ChemDraw reaction file format, but I have ChemDraw open right now, and you can certainly export the ISIS RxN file format. So, with ChemDraw, you could sketch reactions and then use them with MedChem Studio.
Okay, thank you, Dave. A couple of more questions. How long would it take to perform a matched molecular pair screen against a database of 1 million structures?
I was searching 6 million structures in 20 minutes on a 2-processor compound. So within the settings for MedChem Studio, you can specify how many processors you want to use. This one has 2, so I would use 2. So if it's 1 million compounds, and it also depends upon your similarity metric, how high you want the similarity. But I was searching over 6 million compounds in 20 minutes with 2 processors. And I'm sure David could add a little bit more to that.
Okay, thank you, Michael. Another question here is, how easy is it to add new compounds to the MMP database, and how long does that process typically take?
David?
Okay, I think there's a moment of silence here, and you guys can actually hear me. I'm sorry, I'm trying to answer some of these questions, and I think you're not able to hear. But the process of actually creating the screening libraries themselves, it's generally very fast, and I think Michael maybe has a little bit more recent experience. But I think for a 1 million compound database, it's certainly less than an hour. I mean, so a moderately sized database is usually a matter of minutes. So yeah, it's quick to add new compounds, so you can, if there's new structures that come in and you want to add them to the database, it's usually just a matter of minutes. And then that does not need to be changed then. You can screen that database, hundreds of times.
Okay, thank you, David. And what appears to be one final question, what are the advantages of an MMP database screen versus a more traditional similarity screen?
I would say that the the metric we use is much more simple and intuitive to interpret the results. So you just get it's just the number of matches and mismatches between the pair. Things like fingerprints sometimes allow larger and more diverse compounds that it allows us matches. Maybe.
Okay. Anybody else have anything to add?
Not me.
Yeah, I think Michael really nailed it. I think this is really meant to be kind of a counter method to what we call fingerprint methods, which are a little bit more abstract. The measures of similarity aren't mapped concretely onto the molecules themselves. And so, as you saw when Michael was demonstrating the hits, it's very easy because each hit is aligned.
And we lost you again, David.
Yeah, I can see. I think everyone else has to be muted in order for me to be heard. But I think Michael showed it very nicely with the hits, that the combination of the alignment and the highlighting makes it so easy to sort of see which hits are relevant to you versus not so relevant. And with more abstract similarity measures, that just can't be done.
And the nice thing about the matching it by substructures, it puts a lot more context on the smaller features and common features like amides and phenyl groups. In that, if you have a phenyl group in basically the same place, it adds a lot more to the similarity when it should, looking at it as a chemist, versus if it's floating off someplace completely different in the molecule. It will show up in fingerprint similarity, but you really don't want it to.
Okay, thank you, Bob. Thank you, Dave. I don't see any other questions, so what I'm going to do now is just transfer controls of the screen over to Anu, who's just got a couple of closing comments.
Hello?
Yes?
Go ahead, Anu.
Okay. Hi. So I'm just trying to close these other applications. Sorry about that. Okay. So, yes. So you see my screen now?
Mm-hmm.
Okay. So we are going to be at the American Chemical Society in Indianapolis, where Bob Clark will be giving 2 talks. So if you're attending this event, please, that's the place we would be happy to see you. And we will also be in Boston in September. Dr. Michael Lawless is going to be at the Discovery on Target meeting. If you're in Boston and if you would like us to visit your site, or if you're attending the meeting, then that's the place to meet Michael. And I will be in Scottsdale, Arizona for the Drug Discovery Reinventing meeting and I'll be doing a poster presentation there. So if you're attending this meeting, I'd be happy to see you. And also, we offer 2 weeks of trial licenses. So if you're interested in evaluating EdMed Predictor in MedChem Studio, you can reach me. I will be following up with you. Or if you're interested in any kind of support services, then Dr. Michael Lawless will be assisting you with your contract projects. And if you're interested in evaluating GastroPlus and DBD+, then John DeBella is the contact. So as I mentioned, I will be following up after this webinar with the details of the contact, and we'd be happy to assist you in your future needs. Thank you.
Okay, thanks, Anu, and thanks again to Michael, David, and Bob. That is going to conclude today's webinar. Again, if you missed any part of today's presentation, the playback will be available at our website, www.simulations-plus.com. Thanks again for joining us, and have a wonderful day.